There is present in plants and animals a substance which is without doubt the most important of all the known substances in living matter. And without it, life would be impossible on our planet. This material has been named protein. When the chemist Gerald Mulder in 1838 devised the name protein, which means primary, he thought that there was one chemical substance common to all living things that was the essence of life. However, there is not just one substance called protein, but many thousands of varieties of similar chemicals called proteins. But Mulder's intuition was right. Life would be impossible without protein. A major component of all living cells is protein. For example, in a bacterial cell, 70% of its dry weight is protein. Many important proteins have structural functions. For example, in muscle tissue, the contractile fibers are protein. Tendon and other connective tissue are protein, as well as some specialized structures such as hair, feathers, and silk. However, among the most important proteins are enzymes, which control the chemical reactions that maintain life. This is a field ion micrograph of the enzyme ribonuclease, which is about 4/millionths of a millimeter long. The protein molecule is a polymer. That is, it's a large linear molecule that can be split into small molecules. These small molecules are called amino acids. One end has an amino group consisting of nitrogen and hydrogen atoms. The other end is a carboxylic acid consisting of carbon, oxygen, and hydrogen atoms. Amino acids will readily form bonds because a hydrogen from an amino end will combine with an hydroxyl group from the acid end to form a water molecule. Then the two amino acids will form a chemical bond. This bond is known as a peptide bond and is the basis on which proteins are built. In nature, there are about 20 common amino acids differing in their side chains. A protein polymer is a linear assemblage of these different amino acids in a variety of sequences, varying from 50 to several hundred amino acids. The structure and chemical activity of a protein are determined by these side chains. The protein polymer is strongly linked together by covalent bonds. However, the way a protein polymer folds and cross-links to form a compact molecule is mainly determined by weaker bonds. This kind of bond is called a hydrogen bond and is very important to proteins. Cross-linking of protein polymers may occur as a result of hydrogen bonding. The protein polymer is strongly linked together by covalent bonds. However, the way a protein polymer folds and cross-links to form a compact molecule is mainly determined by weaker bonds. This kind of bond is called a hydrogen bond and is very important to proteins. Cross-linking of protein polymers may occur as a result of hydrogen bonding. Acidic or basic side chains will also become charged in a water solution. The acidic side chains will lose a hydrogen nucleus or proton, leaving extra electrons, so the side chain is negatively charged. A basic side chain will gain a proton and thus be positively charged. These molecules that have an unequal distribution of charges are known as polar molecules and have a great affinity for water, which is also a polar molecule. Water molecules are normally hydrogen bonded, and they will form hydrogen bonds with the polar side chains of the protein polymer. If polar molecules, such as water, are mixed with nonpolar molecules, which have no charge, such as oil, the water molecules will clump together and exclude the nonpolar molecules. Nonpolar molecules, such as oil, which separate from water, are called hydrophobic. Some of the amino acid side chains are hydrophobic and are excluded by water. Almost any protein has some of its side chains which are polar, here identified by P, and some which are nonpolar, here identified by N. A protein polymer, which is almost always suspended in water, will naturally fold so that the polar side chains are outside and the hydrophobic or nonpolar side chains are inside, away from the water. This action is similar to the formation of oil drops in water and is the reason why many proteins have a globular shape. This hydrophobic folding of proteins is sometimes strengthened by hydrogen bonding or by covalent bonds between the sulfur atoms of the amino acid cysteine. This bond is known as a disulfide bond. There are two common structural formations in proteins that are caused by hydrogen bonding. Two or more protein polymers may be cross-linked in a sheet known as a beta sheet. The structure of silk consists of layers of beta sheets. These sheets will slip over one another, which gives silk its flexibility. However, the beta sheet will not stretch easily, because this would require pulling against the strong covalent bonds within the protein chains. Another common protein structure is a right-handed helix, known as an alpha helix. This helix forms because of hydrogen bonding within the polymer. The alpha helix is a stable structure that is frequently found in proteins. The structure of hair protein, or keratin, consists of alpha helices cross-linked by disulfide bonds. When hair is pulled, the hydrogen bonds of the helix are broken, then reform again with the help of the disulfide crosslinks. This structure gives wool its tensile strength and elasticity. Keratin and silk are both fibrous proteins. Most structural proteins are fibrous. However, most enzymes are globular. This particular enzyme is trypsin, which digests proteins. This model shows the overall shape of the enzyme. All enzymes have an active site on which the molecule that the enzyme acts upon will fit. This molecule is called the substrate. The side chains of the enzyme that act upon the substrate are indicated in color. The substrate is held in position by one of the side chains of the enzymes. The other side chains act upon the substrate to produce a chemical change. Trypsin is a digestive protein secreted into the intestines to break up the proteins of food for assimilation by the body. The natural substrate of trypsin is therefore another protein chain, and the bond that it breaks is the peptide bond. This reaction is virtually instantaneous. Each enzyme in the cell has an active site which is specific to a particular type of substrate. Almost every reaction in the cell has a specific enzyme to catalyze it. Enzymes function in two ways. They speed up reactions that would otherwise take a long time to occur, and they direct the reactions along specific pathways. Proteins serve two important functions in a living system: as a major component of its structure and as catalysts to control the chemical reactions of life. Life can be defined as a complex, self-maintaining system of chemical reactions. Proteins control and coordinate this system of reactions. Proteins make possible this incredibly complex organization that we call life.